Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss the regulation of calcium by calcitonin. In the last two videos, we discussed how calcitriol, the active form of vitamin D, regulates calcium levels in the serum. And then we discussed in the last video parathyroid hormone. And in this video, we shall look at how calcitonin regulates serum calcium. With the help of calcitriol, parathyroid hormone, as well as calcitonin, the serum calcium concentration is maintained around 8.5 to 10 milligrams per deciliter in a healthy individual. A few words about calcitonin. This is a protein, a peptide, a small peptide, about 32 amino acids. So you can see the molecular weight is only about 3.4 thousand Daltons or 3.4 kilodaltons. This peptide hormone is secreted by the parafollicular cells or C cells present in the thyroid gland. These C cells that constitute a very small part of the thyroid tissue, only about 0.1% of thyroid tissue, also produces, besides calcitonin, other peptides, including one that is called the calcitonin gene-related peptide, or CGRP. As we shall see in a few minutes, calcitonin will bind calcitonin receptor CTR on its target cells, and CTR belongs to the G protein couple receptor family, and this binding of calcitonin to its receptor on target cells will stimulate G-alpha S, that will lead to the activation of adenylate cyclase, causing an increased cyclic AMP that will then go on to activate protein kinase A. It is also associated, the CTR is also associated with G-alpha Q, and that leads to activation of phospholipase C, protein kinase C, which results in the increase in intracellular calcium concentration. Now, calcitonin synthesis as well as secretion is stimulated by the serum calcium levels. So in increasing amounts of serum calcium will cause the calcium to bind this calcium sensing receptor that we talked about under parathyroid hormone. So similarly, these parafollicular cells have CaSR, and this calcium will bind the CaSR, and that binding will trigger intracellular signaling, similar to what we discussed in the last video on parathyroid hormone. So one of the effects of this activation of the CaSR is that the CaSR is associated with G-alpha Q, as we discussed under parathyroid hormone in the last video. And this results in the activation of phospholipase C and protein kinase C that goes on to increase the concentration of IP3 in the cell. And this IP3 then goes to the calcium channels in the endoplasmic reticulum, making them open, and intracellular calcium increases as calcium goes from the ER into the cytoplasm. Now, this intracellular calcium, this high concentration of calcium in the cytoplasm, will increase calcitonin secretion. Okay, so this is unlike PTH. So PTH in the last video, remember, we discussed that although the calcium concentration increases, PTH secretion decreases. So that's very unusual for PTH to do that. Okay, most cases, whenever intracellular calcium increases, the secretion of the biomolecule, whatever it is, a hormone or neurotransmitter, that tends to increase. So PTH was unusual. Now, besides calcium, there are several other biomolecules such as gastrin that are known to regulate calcitonin secretion. Gastrin is known to stimulate calcitonin secretion. The major effect of calcitonin is on the bone. Calcitonin inhibits osteoclast activity. Remember, osteoclasts are the cells that demineralize bone. So that means there is no bone demineralization or reduced bone demineralization or basically resorption. That's another word for demineralization. And that means there is no calcium or phosphate release into the bloodstream. Calcitonin does have a minor effect on the kidney. It basically decreases the reabsorption of calcium and phosphate. This means the excretion, calcitonin increases the excretion of calcium and phosphate. Let's see how calcitonin inhibits bone demineralization. So calcitonin will bind to the receptor, the calcitonin receptor, CTR. And CTR is present in several cells, including osteoclasts. So in the bone, calcitonin will bind to receptors on the osteoclasts. This binding to the receptor, to the CTR receptor, is going to trigger the signaling pathways through G-alpha S and G-alpha Q, and that is going to lead to increased intracellular calcium due to the increase in IP3 that is stimulated by phospholipase C. So there's going to be an increased intracellular calcium in the osteoclast. This signaling will cause the osteoclast to contract. The cell contracts and it loses its ruffled border. So it loses this high surface area that is necessary for this osteoclast to destroy or degrade the bone. This signaling also suppresses osteoclast motility. So the osteoclast cannot move around in the bone to destroy the bone. Then it also inhibits the release of biomolecules that are necessary for degrading the bone. Biomolecules such as acid phosphatase that breaks down organic phosphates, as well as carbonic anhydrase expression. Remember, carbonic anhydrase is necessary to produce protons or acid that is required to break down the bone. So due to these effects, the osteoclast will lose its attachment to the bone and its ability to resorb bone is reduced. And this means without bone demineralization, remember, bone is the largest source of calcium and phosphate in the body. So without demineralization, there won't be any calcium and phosphate coming from the bone. In summary, through inhibition of demineralization as well as inhibition of reabsorption, calcitonin is going to decrease the concentration of serum calcium as well as decrease the concentration of serum phosphate. Before we finish this video, just a few words about another biomolecule called fibroblast growth factor 23 or FGF23. Now this is a protein, 251 amino acids, so molecular roughly about 31,000 Daltons or 31 kilodaltons. So whenever the phosphate levels or the PTH levels or the amount of calcitriol in the bloodstream is high, so under these conditions, what happens is these bone cells, osteoblasts as well as osteocytes, they will secrete this biomolecule FGF23. Now, FGF23 will go to the kidneys and decrease renal phosphate reabsorption. That means it increases phosphate excretion. FGF23 also suppresses the synthesis of calcitriol. FGF23 goes to the parathyroid gland and inhibits parathyroid hormone secretion. So all of these effects by FGF23 is going to cause the decreased levels of calcium as well as phosphate in this serum. So that is all about calcitonin and FGF23. In the next video, we shall discuss hypercalcemia as well as hypocalcemia. So until the next video, once again, thank you very much for supporting this channel. Have a wonderful, wonderful day, and we'll see you soon in the next video.